Bioinformatics.
In this lecture, I'm going to talk about where we actually can find sequence data, both assembled and raw format. So before I get into that, we're now several weeks into the class, and you've probably started to experience this feeling of getting stuck during the labs and running into issues and banging your head against a wall. And I provided some information in the resources tab of Canvas on what to do when you feel stuck, but it bears repeating a number of these things. First and foremost, remain calm, stay positive. It'll get fixed. I've never had someone stuck on one part for the entire semester. So kind of things I generally try to do if I find a command isn't working is I look at how I used the command last. So maybe scroll up. You've probably used it before. And if you haven't, make sure that you typed everything correctly, including both all the arguments as well as the paths. The paths to your files that you're either going to be creating or using is where most of the failure seems to happen as far as executing commands. Next, make sure you're giving the program all the information that it's requiring. Does it want you to explicitly name an output file name? Does it do it automatically? Have you checked out the manual? Have you Googled for examples of people running it? You could very easily find examples of people having run Trim-O-Matic with paired-in reads before, for example, and you could look at what their format was. Have you tried asking somebody for help? That's really a good way to learn, is by working back and forth with someone and teaching them what you're learning and them teaching you what they're getting a better grasp on. And so, another resource, of course, is myself, particularly during the lab period. Don't be shy about letting me know when you're really struggling. I think it's valuable for you to work things on your own first, but there's going to come a point in time, and there's nothing wrong with it, that you need, help from someone that's a little bit more experienced with this stuff. And I can show you maybe where you've been going wrong, or identify if it's something that you didn't do wrong and it's just an error with your server, something like that. Okay, so enough of that. What we're going to be doing today is talking about types of genomic data and where to get them. So all sorts of things we can think about as being aspects of bioinformatic or genomic analyses, everything from raw next-gen sequence data to annotated genes to genomes, variants associated with genomes, functional data, pathways between genes, as well as proteins. You can find a database for just about anything. You probably didn't know that there was a database of genes that are responsive to hypoxia in fish. I didn't either, but there's a whole database on that if that's something you're interested in. I'm not going to talk about all these kind of niche databases, though. I'm going to focus on just some of the big ones that you'll probably run into no matter what your field of interest is at some point or another. This includes DNA databases for the most part, as well as some gene expression databases. But we'll focus primarily today on the big DNA databases. So NCBI GenBank is probably the biggest houser of sequence data in the world, and it's been around for quite a while, since the advent of sequencing. In fact, if you wanted all the known sequences in the world and access to the GenBank database back in 1987, it was actually housed within just several books. It was not actually a website. You would order these books and they would have all the sequences written down in there for you. So we've come a long way from there. Here you can see some of the estimated growth between 1982 and 1998 in the total number of base pairs housed within NCBI GenBank. So they got up to about 3.8 billion by the end of the century. Much like everything else, though, not too long after the year 2000, when next-generation sequencing and whole genome sequencing took off, all of a sudden that has started to outpace the amount of contributing bases to GenBank. So we're far, far above that 4 billion now. There are several different types of repositories within NCBI GenBank, and I want to kind of point out the differences between ones you might run into. So the first one is nucleotide. Now, nucleotide is just a database where pretty much any sequence data is deposited. If you sequence and assemble a gene or a transcript, you can upload it. In fact, most journals require you to upload it. And so you can search by species, you can search by gene name, whatever, and you'll see tons and tons of results. It's not super curated though. There's going to be a lot of overlap. Maybe 500 people submitted sequences for the same gene from the same population of a species. It's okay to have lots of overlap in this database. If you're looking for a really well-curated representative sample of a gene of interest from a given species, you would look within NCBI gene. This is where there should be one representative for any given species. So if you're wondering, oh, I wonder what this gene looks like from mouse, you would go to NCBI gene to look for those nice, well-curated copies. There's a set of criteria for something to be added to NCBI gene. You can go ahead and pause it and read this if you're so curious. As of right now, there are around 70,000 organisms that are represented within NCBI gene. That is not to say that their whole genomes are represented, but they have at least some number of well-curated genes that are represented there. There's over 18 million transcripts and over 84 million different proteins represented. One that I don't think gets looked at or talked about very much, maybe because it's not super high throughput, but I think is a fun exploratory tool, is the Gene Expression Omnibus. So if you go to the Gene Expression Omnibus, you can search for whatever kind of research thing you're interested in, and you'll get a list of experimental outputs. Not only is this the sequence data, but it's actually got some basic visualization and analysis associated with it. So we could think about maybe we're interested in the flu, H5N1 infection over a certain time course in humans and response to different things. So if you were to click on that, you actually can look at gene by gene what the response is in their different samples. So if you're not used to looking at these kind of graphs, here is a timeline. Each one of these is an individual sample, and then whether it was which the treatment was or the control. So here we can see that the treatment and the control at hour 6, 12, and 24 were not different. I don't think there's any stats in here right now. This is just kind of qualitative. We'll teach you later in the semester how to run stats on these types of comparisons though. But we could look at a different gene and go, oh, well, this gene might be involved, because if we look, there is what looks to be some sort of difference between the treatment and the control that gets more pronounced over time. So this gene might be involved in response to that influenza infection. So this is a nice tool if you're just trying to think about research projects and you want to explore and see what we might know or might not know. It's a good kind of fishing expedition. If you're interested in protein, which we don't work with a whole ton in this class, but there are kind of equivalents to the nucleotide and the gene database within NCBI. There is UniProt, which is the equivalent of NCBI nucleotide. Pretty much anyone that sequences a protein can upload it there. It's not well curated, tons of overlap. SwissProt is pretty much the NCBI gene or RefSync equivalent for proteins. Very well curated, non-redundant. And then you've also got some other PFAM where you can look at alignments and relationships between different protein families. If you're interested in protein structure, you can also, there's a database for where protein structures have been experimentally derived via crystallography. And then also there are a number of places if you want to just predict protein structure given the sequence. Okay, but this is a genomics class, so where do we get genome-ish type data? So NCBI has genome. You can search for any given species name you'd like. And here you can look at, there are lots of eukaryotes, prokaryotes, viruses, etc. You can take your pick of any of them that you want. And I'll pick a small one here. This is a abalone shriveling syndrome associated virus. I don't know anything about that, but I do know it's a small genome, so it's easy for us to look at right now. It is 0.03 megabases, so 34,000 bases. What's cool about this is you can see where all the genes are that have been annotated within the genome, and you can actually click on any one of them. And you can click on them, and you can see what it's been annotated for if it's been annotated. You could also pull up a FASTA view of just that gene. You could also blast it and look at other matching results, all sorts of things. We can also just download the whole genome, particularly on these smaller ones, and this should look familiar. Think to yourself, what format is this? This is a FASTA format, right? One line with an identifier. In this case, it's telling you what it is. It's this version of the abalone shriveling syndrome associated virus complete genome. And there's the genome. It's a virus, so it's not multiple chromosomes, so it would be housed within one FASTA sequence. Something like us, where we have many linear chromosomes that are not connected to one another, you would have, one of these potentially identifier plus sequence lines for every chromosome, assuming there's no gaps in your assembly. So the number of complete genome projects is continuing to grow. Something that's happening more frequently, though, is as we get better at sequencing and people start to do lots of sequencing on non-model organisms, is there tend to be a lot of projects that are in perpetual kind of draft, almost permanent draft format, where it gets to the point where it's not worth the time and money investment to make it a perfectly complete genome. This is very true of eukaryotes, where there's lots of complicated, highly repetitive regions. But if 80% of the genome's assembled, a lot of people will say, good enough, we don't need to, dump another $300,000 into trying to finalize that last little repeat region. Here you can see kind of the distribution of completed genome projects. It kind of makes sense that there's more bacteria that have been completed. They're much easier to sequence and assemble. They're smaller. They have less repetitive regions. Eukaryotes tend to be much more difficult. Something else, if you want to kind of really explore genomes of a model organism, is the UCSC genome browser. So they have a number of really kind of well-curated model organisms, things like human, mouse, marmoset, Arabidopsis, all sorts of things. And you can pull up the whole genome sequence for, or in this case, every chromosome. So here's chromosome 15, and we're looking at a little piece of chromosome 15. We can see all sorts of things. We can add different tracks that line up with it. So you could add known gene expression from different tissue types. You can also map it to human or gorilla or orangutan and look at how much homology there is. You can see that this marmoset monkey has much more homology with human, gorilla, and orangutan than it does with mouse, dog, or opossum. Very little with opossum. This is another kind of nice way to explore things. I found an interesting paper where there was a gene associated with cocaine-induced place preference in marmoset monkeys. And I went, well, let's see what this gene looks like on this genome browser. And so here we can see that humans have a full homologous copy and mice have a little bit, but for the most part, none of these other species seem to have a big homology with that gene. We could look at where that gene is turned on by mapping some gene expression data against the genome. So places that are dark are more highly expressed. And so this region of the genome where that gene is located, this over here is a different gene, is actually on in all the brain tissue, but not muscular skeletal tissue very much. And that would make sense. Finally, if you want to download perfect genomes, or as close to perfect as we have, the gold standard is downloading them from Ensemble. This is only for model organisms, so you need to be working on something like human or Drosophila or Arabidopsis. But if you are and you want to download them, they have a very nice, easy system where you can just kind of curl down from their FTP server. And in fact, you've done this in some of our labs before without realizing it, perhaps getting them through Ensemble. So here, this is how you would get the most recent version of chromosome 38 of humans. Now, what we're going to be working with mostly this semester is the SRA. This is the sequence read archive, or I always for some reason remember it as short read archive, but that's not the case. It's where all of the raw data are housed. So if you publish a paper and you're using some Illumina sequencing, most journals require that you make the raw sequence, so what you originally get off the sequencer, publicly available. In other words, you upload all your raw data before you do quality control, before you do trimming. All that gets uploaded to the SRA. Anyone has access to download your raw reads and rerun analyses, hopefully. Here I'm searching for something that's an autotetraploid, and Oryza sativa or rice is an autotetraploid. So some of these previous experiments on rice pop up, and I could go and download some of the data from these experiments if I wanted to. I could click on one, And I could see here is the run number for the SRA, and then the number of bases. So about 500 million bases were sequenced there. I could use that number to download it, and I'll show you how to do that in today's lab. I could also click on their project number and see all the SRA accessions associated with this project. So here we've got diploid rice, autotetraploid rice, different embryonic stages. And so they probably did, without having read this paper, they probably did some comparisons. And if I wanted to redo their analyses with maybe a new version of some piece of software, I could download these data and redo it. Or if I had questions that they didn't address. A lot of people publish papers repurposing already published data. So today you're going to be learning how to data mine the SRA. That's going to be probably one of the most important things you learn early on this semester, because if you ever get into this type of research and you don't want to spend $100,000 on sequencing, you can very much go and answer countless questions with the terabytes of data, the petabytes of data really, that are publicly available that have way more ways to be analyzed if you can come up with interesting questions. You could do research forever for free, basically, if you get really good at asking types of questions and repurposing existing data. Before we go do that, I asked you to kind of skim through a paper last time, and I just wanted you to think a little bit about reproducible science in the context of genome sequence or big data. We need to think about what makes data and research reproducible when you're using next-gen sequencing, and how difficult it is to do those things, because they're, at a certain point, certain things are too difficult and people aren't going to do it. Well, the least difficult thing is to deposit your raw data, right, in the SRA. It's not that difficult to include a summary of your analytical decisions. This should be your methods. A lot of people don't realize that methods aren't just wet lab things. It's not how much you pipetted. It's also how did you clean your raw data? What Phred score cutoff did you use? What size sliding window? What program? Those are all methods that impact your results. And so clearly outlining that is important. If you want to get really in-depth, you could even provide a list of detailed descriptions of all the commands that you used and how you used them. You could also provide some of the intermediate data if you wanted to. That would be more time and take more space on your computer. Something I like to do is when my analysis is all done, is I like to provide a supplementary file that has from start to finish every piece of code that was run to get the raw data into the final version so that anyone can rerun the analysis. And then kind of one of the most difficult things, not really in the sense that it's hard to do, but it's just tedious, is providing all the information about your operating system, software version. Who knows, maybe the version of Trim-O-Matic you used had a bug that in certain file sizes over-trimmed. If you didn't tell the reader that you used that version, they might rerun all your analyses with a newer version, get different results, and wonder why. So as much information as you can provide is very critical. So if you think about that in the context of this really kind of high-profile Ebola virus outbreak paper, how hard would it be for you to reanalyze their data? How reproducible is their analysis? The Ebola is gone. It's what was out there in 2014 is not still out there. You can't go resample it at that time point. Could you access their raw data? If you were able to look and try to find it, it's actually not very clear. It is what I would say is the most important piece of the whole paper is where their raw data are. And it is, in one line, it says sequence data are available at NCBI, Biogroup, PRJ, so they provide the project number. But it's not even in the main paper. It's in the acknowledgments for some reason. But that's good. We can go there. We can see that 249 nucleotides, 891 SRA experiments, and lots of proteins were sequenced. So that's great. Where did those 249 nucleotides and all those protein sequences come from? So they have the raw data, but they also have their, cleaned up and assembled data as well. What methods did they do to get from point A to point B? The method should tell us, right? Well, to find a little bit of this, you have to dig through 29 pages of supplementary info, and all it says is that Ebola virus reads were extracted from the demultiplexed FASTQ files using LASTAL against a custom-made database containing all full-length Ebola virus genomes. So what does custom-made mean? And how could we reproduce that? Should that custom-made database be made available as well, or at least described how to redo it? How did they decide which Ebola viruses' genomes were considered full length? Because they do vary slightly. What arguments did they pass to the last-all command when they were doing the extraction? Because it's not just a one-off thing. There's variations to it. If you wanted some more information, you could download some individual files from them as well, but they're listed in a PDF, which makes them very difficult to process, and some are in Excel, but don't have a consistent format. So this has been made very difficult to actually access in any meaningful way. So that's just food for thought. It's important to think about reproducibility in science. But today what you're going to be doing in lab is you're going to be learning how to access and pull down some of these both curated as well as uncurated and raw sequence data from the SRA, and start to do some manipulations with those as well, which will form the basis of how we get data for the rest of this semester. So as always, I'll be there in lab with you, ready to help.